Hello and welcome to today's webinar. On behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for attending. I'm your moderator, Ellen Capon, Editorial Assistant at Drug Target Review. Today's speaker will be Dr. Carlton Sage. Following his presentation, SAFIRE, AI-predicted ADME for drug discovery leveraging BioPrint, we will move on to a live question and answer session where you can pose questions to Carlton. Please remember you can submit questions at any point during the webinar using the questions panel situated in the menu on the right. So without hesitation, may I pass over to Carlton.
All right, thank you for the introduction. So today I want to talk about several things. We're going to talk about data, models, and AI in drug discovery. Then we're going to talk about the development of SAFIRE, which is our ADME prediction AI tool. We're going to talk about the foundation of SAFIRE, which is the data in a database we call it Emerald. Then we're going to look at how we would have applied SAFIRE to a drug discovery project we ran about a decade ago in a retrospective project analysis, and then talk about next steps. So this is really a story about human scientists, data, and AI that we're going to tell eventually in the context of a drug discovery project that ran from 2006 to 2010. Quickly, a little bit about me. I've been doing drug discovery for a long time now and applying computational tools across a wide variety of projects. Developed a bunch of AI models over the past year, and most recently I spent my time at Eurofins Discovery developing with the team a set of very useful models inside SAFIRE, which is our AI ADME prediction tool. Previously, I've worked on 20+ projects that have brought compounds to the clinic, most recently at Arena, where we put 20 compounds in the clinical trials. 2 of these projects have resulted in compounds which ended up in the clinic. First, Belviq, a drug for obesity, and most recently a compound named etrasimod, which was the reason why Pfizer bought Arena back in 2022. Etrasimod has been approved and is now marketed under the trade name Velsipity. So while at Arena, we worked on, like I said, 20-odd projects. And drug discovery, as you know, if you're in it, and if you don't know, is a very resource-intensive endeavor. And what we did is do a historical analysis of 8 projects, and we looked at from beginning to end, what did it take to get these projects where you can take a small molecule and put it on the clinical doorstep, so preclinical candidate nomination. The average project length was about 3 years. We had some projects that were almost 5 years. The fastest one was just a little bit less than 2 years. The number of compounds we made in every project varied quite a bit. The average was about 1,500 compounds. The high was over 2,000, and the low was somewhere less than 800. And these are compounds that are made, custom made by medicinal chemists for the project, not purchased off the shelf anywhere. And as we all know, drug discovery is expensive. And these prices are from almost a decade ago, so it's only gotten more expensive since then. The average cost to put a compound into clinical trials was $21 million. The lowest we ever spent at Arena was around $15 million to get a compound into the clinic. And as you can see, for each of these projects, they go through a sort of standard ramp up where in the early stages, you have fewer resources. And in the end stages, you're going full steam, and you could have as many as 30 people working in the project at any one time. And the key takeaways from this is that drug discovery is difficult. It's time consuming and it's expensive. And the use of data-based models really, we believe, can make the process more precise and efficient. And I think as we talk about models, it's really important to remember that models aren't perfect, but they can often be very useful. And I want to give some analogies from other endeavors where humans use a set of rules to try to decide what to do best. And so some examples of how we've used models to help us explain the world, right? So the geocentric model. So we used to believe that the Earth was the center of the universe. Well, with the development of technology like a telescope, we discovered that no, the Earth is not the center of the universe, but we actually are part of a solar system that revolves around the sun. Then the idea of how human body functions, this humoral theory that was a way to explain that humans were governed by a balance of these bodily human fluids or humors. We know that's not true today. We keep learning about how complicated this system of being a human is. We also use models to simplify understanding, like the solar system model of atomic theory. It helps us explain atomic behavior and helps us predict things. Second, the human body as a machine. This allows us to break the human body into different functioning systems and look at and hypothesize around these systems interact. So AI has been around a long time, and it was born in the 50s. It went through a period of a lot of research in the 60s and 70s, where we thought we were going to make a lot of progress on applying AI to solve problems that were relevant. We ran into performance expectations there, and there was what we call the AI winter in the 80s and 90s. And then in 1997, Deep Blue beat the world chess champion Garry Kasparov. And that was the last time a human being was the champion of chess. That was really the next resurgence of AI, and it has continued to develop since then. Probably for the drug discovery field, the biggest breakthrough is pretty recent, which was AlphaFold's groundbreaking results in protein folding prediction. And we're seeing it more and more today, used in various industries and a lot in healthcare and drug discovery. And with many technologies, new technologies, they go through this Gartner hype cycle. And I think that we have probably peaked as we move from this, what they call the peak of inflated expectations, as you can see from this article that was recently put out in Endpoints. A lot of the compounds that were AI designed have been put in the clinic, and they've reached their clinical endpoint. Well, they haven't reached their clinical endpoints. They've reached their clinical end because the expectations didn't match reality. And we're getting to the point where we're going to see this probably not too deep trough of disillusionment, because I think we've used these models long enough to realize that we're going to find the practical ways to use them to make drug discovery better. So is hope lost? No, I don't think so at all. So from my perspective, the goal of model development is for predicting any property, particularly in the context of drug discovery. You want to predict or estimate as best as you can given the data you have. And these properties are going to play a critical role in determining the efficacy, safety, and suitability of molecules for potential drug candidates. So when we apply models, how do we think about using them in the context of humans driving drug discovery? So humans are noisy, and I'm highlighting some work from other fields where we use rule-based systems to make decisions about what to do next. And this is highlighted from some work done by Kahneman and Tversky, and some specific examples of that. So if you take insurance policy underwriting, that's something that's super important. You have super highly trained statisticians that develop models for how are we going to underwrite a certain policy to cover a certain item. And Tversky and Kahneman asked, well, how much variability is there between these two experts? Is it 5%? Is it 10%? The industry had never really asked the question before. The answer is that the insurance industry thought that the general standard was 10% variability between two practitioners of that art, when in reality, when they measured it, their variability was 55%, which is a huge deal, not just for the insurance company, but for those of us that need insurance. The second rule-based system is the law. So if you take two judges who evaluated two different cases of a person committing a relatively small offense, cashing a counterfeit check with a value less than $60. How close do you think their sentence times were? Were they within 50%? Were they within 100%? The answer is stunning. One got 30 days in sentence. The other got 15 years. It's a 180-fold difference. Take the law again, where you take 208 federal judges that were given 16 identical fictional cases and assigned the sentences based on their judgment of the cases. And what was the difference between their sentences? The average sentence they gave was 7 years, but the likely difference between their two judgments was 3 years. So 50% error. It's astounding that these systems that are being applied by a human are so biased by sort of the noise that we bring to the table. One thing we can say today is that molecular property prediction isn't to the point of external ballistics prediction. So external ballistics is when you fire a projectile at a target, you know exactly which features you need to know: the velocity, the mass and shape of the projectile, the gravity, the wind, the air density, the spin of the Earth, and the temperature and humidity, so that we can pretty much tell where that projectile is going to end up after it is accelerated. So we did an experiment about a decade ago at a different company than I'm at now, where we were again developing tools to predict ADME properties of molecules. And myself and the head of the medicinal chemistry group decided to get together and ask the question of how well does this AI model do in predicting compounds that it's never seen before, compared to a group of chemists who have never seen the compounds before. So we wanted a pretty fair test, so we needed a relatively large sample size. We had 40 compounds that had new data that hadn't been seen before by the model and probably hadn't been seen by the chemists, but these were known compounds, so if they've been reading their medchem journals, they may have seen them before. And we wanted to predict the bins of permeability of the compounds into high, medium, or low permeability bins. And so we're going to let the chemists work as individuals and as a group by averaging the individuals, predict the permeability bin of the compounds, and we're going to score like golf. So if you get compounds in the same bin, there's no points. Get off by 1, you add 1 point, and if you're off by 2, you add 2 points, and the individual or machine with the lowest score wins the game. So by random, you would get everything compounds in the same bin 33% of the time. You'd be off by 1 45% of the time, and off by 2 23% of the time, with a final score of 36. The best chemists got compounds in the correct bin 55% of the time, off by 1 38% of the time, and we're off by 2 8% of the time, with a final score of 21. The best chemists happen to be also the most experienced medicinal chemist in the group. Take the worst performing chemist, who may have been the best synthetic chemist, so they're not the worst chemist, period. They were off by 0 33% of the time. They were off by 1 45% of the time, and off by 2 bins 25% of the time. And now you ask, how did the average chemist do? The average chemist was in the right bin 35% of the time, off by 1 58% of the time, and off by 2 8% of the time. And how did the model do? Well, the computational model was right in the right bin 60% of the time, off by 1 bin 33% of the time, and off by 2 8% of the time, with a final score of 19, beating everybody, including the best performing chemist. And as we thought about these results, what dawned on us is that what this did, as the computational model did, is it raised the performance of the worst chemist and the average chemist in the ability to predict permeability up to being better than the best chemists, leaving that person to focus their time on things that other areas of the project that they could be working on. So essentially standardizing how they could approach predicting the permeability properties in that way. So our goal at Eurofins in the Discovery AI team is to help our customers make the best decision possible. And I'm going to tell you about the genesis of SAFIRE. So our goal is, given that we can't do the physics yet, and we can't spend the money to test everything, we want to obtain or generate the best data possible. And that's through our Emerald project, which is a database project, and Pixie, which is a project to expand the database of compounds to cover more and more drug-like chemical space. We're going to leverage the data using the best models possible to implementing AI and machine learning approaches, and that's our automated model building project called Diamond. And then make the best decisions possible and be useful to the projects. And when we notice that the models aren't performing as well as they could be, we optimize the models, developing local models, again, using the Diamond tool. So early models of ADME properties seemed so nice. They were simple. So 2 examples here. First is polar surface area, which is a simple calculation of the polar surface area of a compound to predict percent absorption. It's a step in the right direction because you help and explain a problem as a reductionist exercise. However, this is an observation from limited data. And we're going to ask the question, what happens if you expand the data set? So the observation made back in the early 2000s was this polar surface area property. If you could calculate this nice this curve from the data that would correlate directly with the absorption in humans. The second model we've used a lot, and we still use a lot in the industry, is this rule of 5, which is a model of success by association. Chris Lipinski put this together by finding a set of easy to calculate properties, find a simple, easy to remember mnemonic, and use that mnemonic with cutoffs for each property that identify 95% of drugs at that time. So when you move into a larger data set, this is a collection of drugs known in the literature, and ask 2 questions. How did polar surface area do? Well, it's a lot messier than that single correlation line drawn down the middle with the yellow triangles. And then how about rule of 5? Well, if you look at the compounds with the green circles around them, those pass all the rule of 5 alerts, and yet they aren't either super good or super bad. The red circle actually fail all 5 of the rule of 5 alerts, and some of those perform terribly, as you might expect, but some of those actually perform well in terms of absorption. So now we move on to teasing apart our early liability assays from Emerald, this ADME tox collection of assays. So 2 groups broken down in absorption, distribution, excretion, with plasma protein binding, solubility, permeability, efflux ratio, and hERG, and then metabolism, which is metabolic stability measured in human liver microsomes, and then a set of inhibition assays for cytochrome P450s. The Emerald dataset is large. It currently contains about 2,500 compounds that have been tested in all these ADME assays. The dataset is 60% marketed drugs, 5% clinic tested but not marketed, 1% withdrawn drugs, 20% standard pharmacological references, and 12% metabolites in veterinary drugs. The target classes representing the data sets, it's mostly GPCRs with a good collection of kinases, ion channels, and enzymes. And as far as chemical space, shown in this right side graph, we cover chemical space really well. The data set collections we have are large, with the exception of 4 collections. They're all more than 5,000 compounds. For the smaller collections, they're subsets of the Emerald 2500, and they're limited mostly based on the degree of difficulty of those assays. We've also supplemented the data with data from the literature, where we have excellent agreement between the assays tested at Eurofins and the assays published in the literature. When you look at the overlap between the public data and the BioPrint or Emerald data, you can see there's not a lot of overlap for any of these assays. And when there is overlap, we looked at them more closely, and there's a really good correlation between the 2 data sets. So now that we have this great data, we can try to make it into something useful, and that is building SAFIRE. And SAFIRE stands for a suite of ADME tox predictions for in silico refinement and evaluation. And the way these processes work in general is like most drug discovery AI applications. You take molecules as inputs, usually represented as a smile string, and you describe properties for these molecules. They're either calculated properties or fingerprints or other calculated properties. And then you take the combination of the fingerprints, a machine learning method, and the experimental data to find the right combination of descriptors and machine learning method to best predict the experimental properties. We did this for SAFIRE, and we set some predictive thresholds based on a paper published by Baer in 2020, which is a 75% accuracy and a Matthews correlation coefficient, or MCC, greater than 0.4. All of our models pass those thresholds, and our average accuracy is over 80%. We also looked at how well SAFIRE's performance compared to both publicly available tools here on the left. We outperformed 4 out of the 5 models shown here. And if you look at the throughput for the publicly available predictors, they're somewhat limited. You often can run less than 50 compounds at a time, and they're time consuming. And we also compared SAFIRE to a commercially available predictor. and we outperformed it. We wrote all this work up in a paper in Future Medicinal Chemistry. It's a good, quick read that describes how we approached building the SAFIRE tool in particular, and I recommend it for everyone to read if you want to learn more about SAFIRE. We have also released SAFIRE as software as a service. It's got a really simple interface. So you can upload your structures as a CSV file or an SDF file, or just paste them into the interface. Once they're loaded up, you push the Run Prediction button, and your predictions come out looking like this. Easy to read interface. Green means good, red means bad for properties where there are multiple criteria. Yellow means worry a little. We've also put some calculated properties for physical chemical properties like rotatable bonds and log P. And we've implemented some of the more well-known drug likeness property filters as well. The tool is highly performant. For subscribers, you can run 10,000 compounds at a time and get your results back within minutes. We also have released with these how to interpret the data, which is really useful. So if you're just getting started into using predictive tools for drug discovery, and we also enumerate all the physical chemical properties and drug likeness filters and where they're appropriate to be used. So now that we have SAFIRE, we wanted to ask the question of how would it work if we had used it in a drug discovery project like the ones I described earlier in the presentation. And this is the first of what I hope to be several retrospective analyses of drug discovery projects we'll be able to do using SAFIRE. This was for a project looking for ligands for the ghrelin receptor. Agonists of the ghrelin receptor are potential treatment for degenerative diseases. And inverse agonists or antagonists of the ghrelin receptor are potential type 2 diabetes therapy as indicated by ligand and receptor knockout studies showing demonstrated weight loss and improved glucose control. So we approached ligand discovery for this project like most projects. We did an HDS, we did virtual screening, and we did classical medicinal chemistry based on the compounds that were available in the medchem literature. Now, one thing I want to point out is that this project isn't different from the vast majority of projects we worked on in the past. Outside of potency, in terms of experimental measurements, the data is really sparse. So what I've done in this slide is plot the number of compounds in blue and the number of experimental measurements for those compounds in the orange, gray, yellow, and blue and green colors. As you can see, we do really well at measuring potency of compounds. That's one of the major drivers of projects. But we don't really test compounds in terms of these liabilities that can totally knock compounds out of their running for the clinical trial, just as much as a poor potency can. We dug further into the data, and this graph is a little complicated. What we've got is data by year, and in the rows are experimental measurements. And so every yellow bar is a different year, starting in 2006, going to 2010. And then the vertical axes are weeks of the year that we tested compounds. So every week you test compounds and you see how the project is going in terms of the representatives of your medchem sets at that time. And you'll notice that first year, 2006, the project is just getting started. You also notice these orange bands. So the cutoff for potency was roughly 20 nanomolar. And you can see we weren't meeting the threshold for most of the projects, for most of the compounds of the project in the first year, and even as we got into later. You'll also notice that there aren't a lot of horizontal rows filled in, and those are again where we haven't made measurements in the project. Eventually, as we work down through 2009, we get compounds where you have potency and you have metabolic stability and good hERG, which are the other two properties that are highly populated in this slide. You also notice that as we get to the end of 2009 and the beginning of 2010, the number of compounds made have slowed down. We started to test compounds in animals, and we're starting to see that maybe there's not enough oomph in this receptor to be a useful tool for type 2 diabetes. And so the project wound down before it had a chance to hit the clinic. Nonetheless, this is a really great example of how projects typically go. As you can see, you ramp up in terms of number of compounds generated in the first 2 to 3 years. They start to go down as you get closer to making a decision about what to do with the project, and whether or not it's going to work in your animal models and would be a viable potential drug candidate. You also notice that we started adding the metabolic stability and hERG as we got further along in the project, and noticed that it was indeed starting to be a problem. But you could also argue that we could have tested more and maybe made some changes to perform better along that line. So I wanted to take a couple medicinal chemistry series from this project and do a further dive and look at how, if we had SAFIRE at the time, how we could have used it. So this indazole series 1 contains around 727 compounds. And on the left-hand graph is just the measured potency. You can see we have some compounds that are getting into the low nanomolar, into the nanomolar range in that graph. And on the right-hand graph with dots, you have by year the SAFIRE predicted liabilities, where in this case, a 1 would mean you pass all the SAFIRE models, and a 0 would mean you fail all the SAFIRE models. And then we look a little closer at the two attributes that we've experimentally measured. The first is hERG. And in this case, the SAFIRE predictions wouldn't have been useful. The chemical space of the model isn't the same, or the chemical space of the indazole series doesn't match that of the model. But if we've been using SAFIRE, we would have known that from the beginning. We would have known to test more frequently and to build local models. Metabolic stability, on the other hand, the SAFIRE predictions were really useful. So we could have tested a little less frequently, although still test to ensure that we're still in the right chemical space. Also build local models so our precision of predicting metabolic stability would be higher. Second series, this diaminopyrimidine series 2, smaller series, not getting into the right potency range. The liability profiles from predicted SAFIRE are indicating that there's some things to work on. In this case, the hERG predictions were really useful. This means we could have tested a little bit less frequently, again, continuing to test and add in to build local models for hERG. And for metabolic stability, the SAFIRE predictions were useful again, allowing us to select compounds to make and maybe to test less frequency, but still build local models. Speaking of local model development, how do we think that would work? It's going to work very much like our models, larger models, where you take either Emerald data or project data, feed that into a data preparation module. That prepared data then goes into our model iteration mechanism, where we iterate through all the potential combinations of descriptors and machine learning methods, looking at each potential model and asking whether or not it's going to be performant enough to become an optimized model and deploy in our drug discovery pipeline. So that was a brief walk through how you could apply SAFIRE in projects and how we're going to be applying SAFIRE in projects as we move through drug discovery and our integrated projects at Eurofins. And now I'd like to thank the people that really did the work. First, the Discovery AI team, who are shown on this slide. We've got a great group of people from machine learning engineers, to software engineers, to database specialists and project managers that really work well together as a team and have made some really amazing progress over the last year and a half. The Eurofins discovery team as a whole, outside teams from Eurofins come in and help make suggestions on how we could implement and make our tools better, and we super appreciate that. And then finally, the ARENA and Beacon Discovery colleagues from over the last 2 decades. The data sets we have are amazing to work with. And then final thoughts. Drug discovery is hard and expensive. Data is really, really important. And that's why we put together Emerald and Pixie, the project to improve and expand the Emerald database. AI models, when you apply them regularly, you update them and check whether they're working for your chemical space can be really useful. That's SAFIRE and Diamond. And Eurofins has the tools, assays, and experience to help you be successful in your drug discovery endeavors. And finally, thank you for your attention.
Thank you, Carlton, for your excellent presentation. We now have the pleasure to introduce our question and answer session. Don't forget you can still submit your questions using the questions panel in the menu on the right. So to begin, why don't drug discovery projects just measure molecules in all the relevant experimental systems?
So that's a great question. Most of the reason is when you do drug discovery, you kind of have a finite cost or a finite amount of financial resources to get a project done. That includes both making compounds and testing compounds. So projects try to be efficient in the use of that financial resource. The other key is that it takes time to measure those properties, and often the synthesis timing and the measurement timings get out of sync, and you often have to wait a long time to actually make decisions about the data you get back. So using these predictive models can really help you sort of predict before you make.
Where in the drug discovery process is SAFIRE most useful?
So I think early in drug discovery is the place where SAFIRE would be most useful. Taking results from an HTS or a virtual screen, ranking them in terms of their potential liabilities when you're analyzing and look for scaffolds to work on. And that also includes moving into lead identification and then further into lead optimization, where you'll have the opportunity to have internal data and build these local models that I was referring to at the end of the presentation.
Does Eurofins prosecute integrated discovery programs? Is SAFIRE included in these programs?
Yeah, absolutely. We have an entire team that called Discovery One that essentially will come, and if you have a target and an idea of how to approach that target therapeutically, we'll help you find hits for that target, optimize hits for that target, and SAFIRE is part of that whole process.
Is the data set and AI approaches used in the building models going to change?
I think the data set is certainly going to change. We have a project internally at Eurofins called Pixie, which is designed to scour the literature for important compounds. That includes compounds that have been approved or are winding their way through the clinic or have actually fallen out of the clinic. We're going to obtain those compounds, measure all of the important properties of those compounds internally, and then add them to the Emerald data set and rebuild the SAFIRE models so they'll be more useful.
What are the main challenge or the major challenges that we are experiencing with AI in drug discovery, especially AI accelerated virtual screening?
So the major challenges I think that that we're facing is, well, a lot of them are being solved. Access to synthetic space is probably one of the bigger challenges. We know these generative models are great for coming up with ideas for projects, but often having that idea of a chemical turned into the reality of a synthesis is a challenge we struggle with. Also, the vastness of chemical space often can bite us. Models like SAFIRE are general models, and when you work into a localized chemical space, if you're not well represented in the space of compound present in the model you've built for whatever property you're trying to predict, you'll often not do well. So the development of local models and fast turnaround AI model development is another place where we're trying to optimize.
And do you get AI hallucinations? How do you mitigate them and verify outcomes?
Yeah, I think we need a little bit more clarification on what an AI hallucination is.
Hopefully, whoever submitted the question will clarify that for you. Just give them a second.
Okay.
We haven't got a message through yet.
Yeah, I mean, it could be that a hallucination is, the belief that your models are predicting as well as you hope they are. And I think, well, for me, the best way to test those kind of hypotheses is actually to do the experiment and test how well your models are doing on your real project data.
We've got a message through now. So in an AI system, hallucination occurs when the system provides a response with a high degree of confidence, but this response is not supported by the data on which the system was trained. As we wrote in our previous article, this phenomenon can be compared to what happens when a person is under the influence of drugs or alcohol and perceive things that are not really there.
Yeah, I mean, I think my answer fits with that. You have to do the experiment and adjust your approach to model building if you wind up in a space that you clearly don't understand.
Absolutely. So unfortunately, that's all we have time for today. Please join me in thanking Carlton. As you leave the webinar today, a survey will appear on your screen asking you to rate the webinar. Please take a moment to provide your feedback. If now is not a good time, the survey will be sent to you shortly via email. If you could complete it when you can, we would greatly appreciate it. On behalf of Drug Target Review and Eurofins Discovery, I'd like to thank you all for attending today's webinar. We hope to see you again soon. Goodbye.